Hello viewers, welcome to my channel. In this video, we will discuss about some important questions on DNA replication. For CSR NET, ICMR NET, GATE, NEET exam, the questions regarding this topic is very important. Some brief notes regarding the questions and their related options are also provided here, which will be beneficial for you. So stay tuned in this video. The first question. During DNA replication, two strands are separated by Option 1 DNA polymerase III, Option 2 DNA gyrase, Option 3 helicase, and Option 4 is reverse DNA gyrase. And the correct answer is Option 3 helicase. The main function of helicase is to unpack an organism's genetic material. Helicase are the motor proteins that move directionally along the nucleic acid phosphodiester backbone, separating the hybridized nucleic acid strand using energy from ATP. So the helicase use the energy ATP to break down the hydrogen bond between the nucleic acid bases. This process is characterized by breaking hydrogen bonds between the annealed nucleotide bases. They also function to remove nucleic acid associated proteins and catalyze homologous DNA recombination. So next is the DNA Pol III is the primary holoenzyme that involved in the replication activity. It's synthesizing DNA in 5' to 3' direction. DNA Pol III is a component of the replisome which is located at the replication fork. DNA gyrase or type 2 topoisomerase is an essential bacterial enzyme that catalyzes the ATP-dependent negative supercoiling of double-stranded closed circular DNA. They reduces topological strain in an ATP-dependent manner while double-stranded DNA is being unwound by elongating RNA polymerase or by helicase in front of the progressing replication fork. Whereas reverse gyrase is a type 1 topoisomerase that introduces positive supercoils into the DNA. That is in contrary to the negative supercoils introduced by the type 2 topoisomerase, that is DNA gyrase. These positive supercoils can be introduced to DNA that is either negatively supercoiled or fully relaxed. Next question. During DNA replication, DNA strands are synthesized in Option 1 5' to 3' direction, Option 2 3' to 5' direction, Option 3 both of the above, Option 4 none of the above. And the correct answer is Option 1 in 5' to 3' direction. DNA is always synthesized in the 5' to 3' direction, meaning that nucleotides are to be added only to the 3' end of the growing strand. Because the enzyme DNA polymerase has the ability to incorporate nucleotide at the 3' end of the DNA strand. This happens because DNA polymerase requires hydroxyl group present at the 3' end to catalyze the phosphodiester bond formation. Next question. Termination of replication is triggered by Option 1 SSB, Option 2 is helicase, Option 2 TAS protein, and Option 4 is DNA polymerase. And the correct answer is Option 3 TAS protein. TAS, also known as termination utilization substance, is a protein that binds to terminated sequences and acts as a counter helicase when it comes in contact with the advancing helicase. The bound TAS protein effectively halts DNA polymerase movement. TAS helps end replication in prokaryotes. Next question. During DNA replication, Okazaki fragments elongate Option 1 lagging strand toward the replication fork, Option 2 lagging strand away from the replication fork, Option 3 leading strand towards the replication fork, Option 4 leading strand away from the replication fork. And the correct answer is Option 2, that is lagging strand away from the replication fork. The replication of lagging strand is discontinuous, with short Okazaki fragments being formed and are joined together. The lagging strand is synthesized in the 5' to 3' direction, that is opposite to the direction of fork movement, whereas leading strand is also synthesized in the 5' to 3' direction, but towards the direction of the replication fork movement. Next question is, the nucleotides are joined by option 1 phosphodiester bond, option 2 phosphotriester bond, option 3 glycosidic bond, and option 4 is hydrogen bond. And the correct option is option 1 phosphodiester bond. The phosphodiester bond is the bond between the sugar base of one molecule and the phosphate group of the adjacent nucleotide. And these nucleotides are the building block of DNA or RNA. Next question. The nucleotide consists of option 1 sugar plus phosphate plus amino acid, option 2 sugar plus phosphate plus nitrogenous base, option 3 sugar plus nitrogenous base, option 4 amino acid, phosphate and nitrogenous base. And the correct answer is option 2, that is sugar, phosphate plus nitrogenous base. Nucleotide is the monomeric unit, that is the building block of DNA and RNA. It consists of a sugar molecule attached to phosphate group and a nitrogen containing base. The sugar molecule in case of DNA is deoxyribose, whereas in case of RNA it is ribose. The bases used in DNA are adenine, cytosine, guanine and thymine, whereas in case of RNA the uracil is replaced with thymine. Next question. Which of the following enzyme helps in opening of the double helix in front of the replication fork? Option 1 DNA polymerase 1, option 2 DNA polymerase 2, option 3 DNA gyrase, and option 4 is Topoisomerase 1. And the correct answer is option 3, that is DNA gyrase. DNA gyrase or simply gyrase is an enzyme within the class of topoisomerase and is a subclass of type 2 topoisomerase. It reduces the topological strain in an ATP-dependent manner while double-stranded DNA is being unwound by elongating RNA polymerase or by helicase in front of the progressing replication fork. This DNA gyrase enzyme causes negative supercoiling of the DNA or relaxes positive supercoils. Whereas the DNA Pol II possesses 4 enzymatic activities, such as DNA Pol II possesses 5' to 3' DNA synthesis capability. It also has 3' to 5' exonuclease prepreventing activity. DNA Pol II interacts with multiple binding partners common with DNA Pol III in order to enhance its fidelity and possessivity. DNA Pol I possesses the following enzymatic activities. A 5' to 3', that is forward DNA dependent DNA polymerase activity requiring a 3' primer site and a template strand. The 3' to 5' reverse exonuclease activity that mediates proofreading. The 5' to 3' forward exonuclease activity mediating nick translation during DNA repair and primer removal. So DNA Pol I is involved in nick translation. A 5' to 3' forward RNA dependent DNA polymerase activity. Pol I operates on RNA templates with considerably lower efficiency than does DNA templates, and this activity is probably of only limited biological significance. In a double-stranded DNA, the two strands are joined by option 1 hydrogen bond, option 2 covalent bond, option 3 ionic bond, and option 4 is none of these. And the correct option is option 1 hydrogen bond. The two strands of DNA molecule are held together by hydrogen bonds formed between the complementary nitrogenous base of the two strands. Adenine pairs with thymine by 2 hydrogen bond, whereas guanine pairs with cytosine by 3 hydrogen bonds. That is, A paired with T by 2 hydrogen bond and G paired with C by 3 hydrogen bond. What is the reaction in DNA replication catalyzed by DNA ligase? Option 1, addition of new nucleotide lagging strand, addition of new nucleotide to the leading strand. Option 3 is formation of phosphodiester bond between the 3' OH of one Okazaki fragment and the 5' phosphate of the next nucleotide of the lagging strand. 4 option is base pairing of the template strand and the newly formed strand. And the correct answer is formation of phosphodiester bond between the 3' OH of the one Okazaki fragment and the 5' phosphate of the next on the lagging strand. Next question. Semiconservative DNA replication was first demonstrated in option 1 E. coli, option 2 Drosophila melanogaster, option 3 Saccharomyces cerevisiae, option 4 Streptococcus pneumonia. And the correct option is option 1 E. coli. Messelstahl and Stahl experiment in E. coli in 1958 was an experimental proof for semi-conservative DNA replication. Next question. How many replicons are found in E. coli? Option 1, option 2, option 3, option 4 is 5. And the correct answer is option 1. E. coli is monorepliconic and have single replicon, while eukaryotes contain multiple replicons on a single chromosome and are called multirepliconic. A replicon is the entire region of DNA that is independently replicated from a single origin of replication. A bacterial chromosome contains a single origin and therefore the whole bacterial chromosome is a replicon. The chromosomes of archaea and eukaryotes can have multiple origins of replication, so their chromosomes may consist of several replicons. Next question. Which of the following would relax negatively supercoiled DNA? Option 1 DNA gyrase, option 2 type 1 topoisomerase, option 3 DNA helicase, and option 4 is ligase. And the correct answer is option 1 DNA topoisomerase. Negatively supercoiled DNA can be relaxed by topoisomerase 1. It creates the nick in one strand of the DNA and rotates freely to resolve supercoiling. Another important thing is that this type 1 topoisomerase is also known as reverse DNA gyrase. Okay. So type 1 topoisomerase is also known as reverse DNA gyrase, whereas DNA gyrase, simply DNA gyrase, is known as type 2 topoisomerase. Next question. If the template DNA strand has nitrogenous base sequence ATGGCC, then the newly synthesized DNA will have option 1 UACCGG, option 2 TACCGG, option 3 TAGGCC, and option 4 is ATGGCC. And the correct answer will be TACCGG. In only one of the two DNA strands serve as a template for real replication. The antisense strand of DNA is reached by DNA polymerase from 3' end to the 5' end during replication. The complementary DNA is created in the opposite direction, in the 5' to 3' direction, matching the sequence of the same strand. The DNA produced during replication is complementary to the template DNA strand. So this directionally is because DNA polymers can only add nucleotides to the 3' end of the growing DNA chain. Here it is mentioned that the ATGGCC is the nitrogenous base sequence in the template strand, and you have to find that the sequence of the newly synthesized DNA. Here it is mentioned that DNA produced during replication is complementary to the template DNA strand. So the complementary A is T A G C, again G is C, next is C G C G. So TACCGG. Okay. Next question. In a molecule of double-stranded DNA, the amount of adenine present is always equal to the amount of option 1 guanine, Option 2 cytosine, option 3 thymine, and option 4 is uracil. And the correct answer is option 3 thymine. Because in purine and pyrimidine bases should be present in a 1:1 ratio in DNA of any organism. The base pair rule is another name of Chargaff rule. It specifies that the amount of guanine and cytosine be the same, while the ratios of A and T are the same. And A always paired with T by 2 hydrogen bond and T always paired with C by 3 hydrogen bond. In DNA, the enzyme which breaks the hydrogen bond is option 1 ligase, option 2 helicase, option 3 topoisomerase, option 4 is polymerase. And the correct option is option 2 helicase. DNA helicase catalyzes the disruption of the hydrogen bonds that hold the two strands of the double-stranded DNA together. This energy requiring unwinding reaction results in the formation of single-stranded DNA required as a template or reaction intermediate in DNA replication, repair and recombination. Suppose these are the two strands. This is 5' end, this is 3' end. Again this is 3', this is 5'. And these two bases are joined by, this is the hydrogen bond. Suppose it is, again. This is A, this is A, this is T, this is G, this is C, this is C, this is G, this is the 5, and this is the 5' end, this is the 3' end. Again this is the 3' end, this is the 5' end, and these are the hydrogen bond, and these hydrogen bonds are breaked by this helicase and it produces two single strand. Which of the following protein does not involve in the initiation of replication? Option 1 DNA, option 2 is SSB, option 3 DNA B, option 4 TUS, and the correct answer is option 4 TUS. In eukaryotic replication, more than 20 proteins take part. Out of that, the initiation of replication takes place in presence of helicase, primase, SSB, DNA A, DNA B, DNA C. What is origin of replication? Option 1 site at which DNA replication start. Option 2 site which prevents initiation of replication. Option 3 site at which DNA replication terminated. Option 4 random location on DNA. And the correct answer is option 1 site at which DNA replication start. Now, origin of replication is a particular sequence in a DNA genome at which replication is initiated. So, thank you. Please like, share and subscribe to this channel.